So this unit is going to be about microbiology. We'll be talking about pathogens of all sorts, microbes of all sorts. Today's lesson is going to be primarily about viruses. And so let me just get right into that. A virus, as Peter Medawar, famous scientist, said, is a piece of bad news wrapped in a protein. And as you'll see when I begin to talk here, okay, that viruses are just really basically that, okay? The definition of a virus, a piece of DNA or RNA. We talked about that earlier, genetic information. And so really all it is a piece of information packaged up in order to transmit that information to some host cell. The protein is known as a capsid. The protein covers the DNA, protecting it. Also, the protein contains structures known as receptors that can bond to different cell proteins so that the cell will then recognize the virus and pull it inside and allow the virus to go about its business, which we'll talk about shortly. Scientists kind of have wondered for a long time where do viruses belong in the big scheme of things? Are they life? Are they non-life? Scientists don't really feel like they meet the criteria of life. And there's a number of reasons why. First, viruses don't metabolize. They do not use energy. They don't need to be fed. They don't require any food. And so that's one criteria of living things that we talked about earlier this year that they do not meet. They don't respond to the environment. Viruses do not do not change, whether or not it's hot or cold, whether or not it's an acid environment, whether it's shady or sunny. They don't respond in any way to the environment that we would recognize a living cell doing. They don't grow or develop. You can keep viruses in a test tube for years and years and nothing will change with regards to them. And therefore, viruses are not going to grow as we would recognize a living thing grow. And finally, they don't reproduce. The basic definition of reproduction is producing new organisms from the original parent organism. And viruses aren't able to do that. Their basic prime directive is to get a host cell to make copies of itself, which it can do, quite well. And so viruses are not able to reproduce. And again, because they don't have all of these criteria that living things have, no metabolism, no response mechanism, no growth, no reproductive method, scientists don't really believe that they would be classified as living organisms. Viruses come in a variety of different shapes, from the icosahedron of HIV to the long filamental Ebola virus, probably the most notorious hemorrhagic virus. It attacks the lining of your vascular system, your blood vessels, causing you to bleed and allowing you a means of getting transmitted from one host to another. Some are spherical, like herpes. Other are more pill-shaped, like smallpox. Again, the flu virus is spherical in nature, allowing it to live in your air and lungs, your trachea, your lungs, and West Nile, again, also another example of a spherical type of virus. This one, though, not transmitted through the air, but through vectors such as birds. One important thing to understand about viruses is they are not picky in terms of who they seek out for hosts. This one is called a bacteriophage because it basically is designed for attacking bacteria. And here's an example of that where these T4 have latched onto this E. coli that you can see in the center there. They've basically short-circuited the E. coli DNA, and basically that E. coli has just created many, many, many viruses, so much so that the E. coli eventually burst open, releasing all those viruses that can now basically find new hosts, which could be used for transmitting that virus even further. Some are spiral shaped, like this tobacco mosaic virus. Can't really tell that in this picture, but in this picture here, you've got all these tobacco mosaic virus. They're very long spiral shaped ones. And again, I point this out because this is one that attacks plants. We have concerns about this one at times because of how it can be harmful to our food crops, tomatoes, cucumbers, squash, peppers, etc., as well as ornamental plants. And so it can play a role in harming our food supply. The point I'm trying to make here is that, while we are most familiar with the viruses that attack us, HIV, Ebola, the flu, there are viruses that can live in any living organism. So now I want to just begin to talk about how viruses can cause disease and why we're so concerned about them. Scientists recognize that there's two methods of infection. Both work in a similar in a similar manner. Before I get into each of those, I'm going to tell you or suggest strongly that you pause the video here and watch the brief video how a virus works. It gives you an overview of the general strategy for how viruses operate inside your body. Once you do that, then come back to my notes and listen further, and I'll talk about, again, the two methods by which viruses can operate inside the human body. Lytic method. The root lyse means to dissolve away, and so the lytic method, involves a virus entering the cell and short-circuiting the genetic information inside that cell, getting the cell to immediately start making copies of the virus. And so literally the cell becomes a virus-making machine. It fills up with viruses, and eventually those viruses will, burst out of the cell and travel through the environment to find similar cells with similar receptors that they can infect. This would be the sort of quick and dirty method of infection where you're infected relatively quickly. The best examples that you might be familiar with, or perhaps I should share, are flu and Ebola. You get infected with the flu, within 24 to 48 hours, you feel pretty miserable. With Ebola, again, within a week to 10 days, you are nearing death. You're bleeding from your various orifices, nose, ears, mouth, hemorrhaging blood, and in a short time you're dead. So both of these viruses work in that lytic method where they're basically quickly destroying your cells. The flu doesn't quite kill you, but it does make you feel weaker. It can kill you. And what it does, you can tell that you have the flu from your change in temperature, from the mucus builds up in your lungs, etc. But you know that you're sick very quickly. So this is just one more visual to try to help you understand how, the cell absorbs the virus turns off the cell, the virus gets the cell to make lots of copies of that virus, and then the virus is released when the cell bursts open, and these viruses can now find other host cells to do that same process. Again, lytic because it happens relatively quickly and doesn't take very much time for it to occur. The other lifestyle or infection method is the lysogenic. Again, it's got the word lyse in there, so cells are destroyed. Genic kind of implies that this is taking place over the lifespan of the cell or the organism, and so this would be more like the drippy faucet method. In this case here, what makes this one a little bit different is the virus's DNA becomes incorporated actually into the cell's DNA, and so as new cells are made, the cells that are being created contain that virus DNA, and every time a new cell is made, again, information for making that virus is also transmitted to that new cell. And so in some cases, Viruses will get made periodically as pieces of the DNA are read and proteins and such are put into play. At other times, the viral DNA will be read, and then all of a sudden another protein will be read, and you'll see it go through a lytic stage as well. But this can take place over a long period of time, and so you don't necessarily always know that you're infected if you have a viral infection that's like. And so, two examples: HIV and herpes. Again, both cases, they will infect the host cell and just begin to slowly create new virus. And this can go on for years and years and years. And you may not recognize that you have it because you may not show any symptoms. You may not show symptoms for a period of time until you have enough of what's called a viral load for those symptoms to have an impact on you. And so lysogenic is one that, I think is a little bit more scary, dangerous, because, it kind of stays hidden for a while. As you can see in this visual, the DNA gets incorporated into that cell DNA. You can see that red DNA built in there, and every time you get new cells being produced, then it's got the ability to create new viruses. In this visual, it goes through a lytic stage, and so all of a sudden, some trigger will tell the cell to suddenly make a bunch of those viruses, and all of a sudden, after a period of time, the person is seriously infected and shows symptoms of that virus, that infection. So two methods. Okay, the quick and dirty method, lytic method, you're infected very quickly. You feel the effects of that infection, such as the flu. Lysogenic, it can, the drippy faucet method, and it can exist in your body for long periods of time before you show any symptoms of the virus. Now, all that being said, we do have some tools for protecting ourselves from viruses, and both man-made and natural. The first one is we have what's known as white blood cells. Those are, the scientific word for those are phagocytes. And phagocytes are able to surround and destroy viruses by eating them. And when that happens, okay, the virus is broken down by enzymes inside the white blood cells, as you may be familiar with their common term, but phagocytes is a scientific term. These are very effective. Problem is they have to seek out and destroy them, the viruses, until they can find them and attach themselves to them and absorb them. You're infected. So it's a solid method, but it takes a while for it to work. Another method that works in conjunction with phagocytosis are antibodies. These are molecules that your body makes that can neutralize a pathogen. As I mentioned before, viruses have receptors that can recognize molecules on the surface of cells. Antibodies are molecules that are created that recognize these receptors because they are foreign to your body, and what they do is, they will then attach to them and neutralize. And they will, as you can see in this visual, the antibodies attach to those receptors on the outside of a cell, in this case a red blood cell, that doesn't belong in your body. We'll talk more about blood types later. But by attaching itself to the proteins, which are referred to as antigens on the outside of the body, this prevents the virus from doing its job, which is to get inside your cell and to get that cell to make copies of itself. So antibodies are a second and really worthwhile tool for helping you protect yourself. The negative of antibodies is you can't make them until you've been invaded by that viral pathogen. The positive about antibodies is once you know how to make them, you're protected for the rest of your life. And so your body always remembers, your body's immune system always remembers how to make antibodies for that virus should you ever be invaded by it in the future, at some point in the future. And scientists, recognizing the value of antibodies and realizing, the harm that can be done if you are infected and you can't protect yourself fast enough, began to investigate, was there a way for us to have protection from a pathogen, a viral pathogen or any other pathogen before we were infected? And that's where the idea behind a vaccine came into play. Vaccines, of which you've all received multiple vaccines over the course of your life, many of which to just give you the opportunity to come to school, is a man-made tool that we've developed whereby we are able to give the human body immunity from a pathogen before you are actually invaded by that pathogen, before it can cause any harm to your body. This was first done back around 1796 when Edward Jenner was able to show that he could protect an individual, in this case a young boy, from the horrific pathogen known as smallpox. And what he did is he basically infected the boy with a mild version of cowpox. And when smallpox reared its ugly head later on, this young boy was then actually protected. This was what we recognize today as the very first type of vaccination. At this point, I recommend that you pause this and go listen to the brief video called How Do Vaccines Work, which is also linked on my website. It gives you a really good idea of how a vaccine does work. Anyway, there are two ways that we can give immunity. The first way is called active immunity. And with active immunity, an individual receives actually the ability to make those antibodies. What happens is, as you saw it in the video, either a dead version with the DNA removed or a weakened version of it, as in the case of cowpox, either one of which, those are both injected into the individual, and what that does is it triggers them to create the antibodies. And as I mentioned earlier, the ability to make antibodies is something your body remembers for the rest of your life, and so you're given permanent immunity to those. Again, a number of different vaccines do that. The one that you got for hepatitis B, the one for mumps and measles, same thing. And so what you're given is this, by getting a natural weakened form or dead version, quote unquote dead version, where all you're getting is the protein coat with those receptors, you're able to protect yourself for the rest of your life. Now, the vaccine is one that gives you passive immunity. It's the term passive. Scientists use this term because all you're getting is the antibodies. You're not being taught, your body's not being taught how to make the antibodies. And so this is the best example of this is what you're familiar with is your tetanus shot, designed to protect you from something known as lockjaw, a disease where your muscles basically seize up and no longer function. But what happens when you get your tetanus shot about every 5 years is that you are getting the antibodies to keep yourself protected from lockjaw, but your body doesn't know how to make them. And so over time, those antibodies are degraded, and your doctor needs to give you, again, a form of passive immunity, gives you a booster shot, which again just adds those antibodies, back into your body to keep you protected for, again, another number of years. Mentioned to you is that there are actually what are known as antiviral drugs. And these drugs are drugs that are able to neutralize a virus, in some ways mimicking how an antibody works, and they work two ways. One group of drugs are able to actually coat the outer cell, the outer covering of the cell, the cell membrane where those receptors are. And what they do is they prevent viruses from attaching to those receptors. And by covering up those receptors, viruses are not able to get inside the cell in order to initiate that copying mechanism. The other type or class of drugs are ones that actually cover the virus. And so they cover those proteins that are on the outside of the virus so that there's no way for the virus to actually attach itself to a cell. So they work very similar to antibodies, but again, these are man-made drugs that either attach to the cell to keep the virus out or attach to the virus so it has no means of attaching itself to the cell. Scientists are continually looking for new ways to, again, protect ourselves from the hosts of virus that can harm us. And these are, again, just a number of these, from the cells that, are natural defenses with the cells that eat us, to the antibodies we make, to the vaccines that we've developed, and the antiviral drugs. So just a brief overview again here of what viruses all about.